The advantages of sexual reproduction can be divided in two categories. Those stemming from the recombination and fertilization of genetic material between individuals, and those stemming from the production of seed propagules. Increased population genetic variability increases the population's adaptive potential and thus its capacity to withstand environmental changes which will inevitably occur. Imagine this is a population of genetically uniform clones of a plant, and that all of these clones are susceptible to plant rust caused by a pathogenic fungus. If the population is attacked by rust, all individuals will succumb to the pathogen because they're all sensible to it. On the other hand, if the population is genetically variable, some individuals will be resistant to the pathogens and others will not. This guarantees that at least some of the individuals will survive when this particular pathogen invades. But also, the population will be resistant to a wide array of pathogens. The increase in genetic diversity occurs because of the genetic recombination which happens during meiosis. Chromosome segments are being shuffled and exchanged between homologous chromosomes, producing new genetic combinations. In here we have a heteroduplex that was produced by the recombination of the blue and red alleles. Furthermore, during fertilization, two gametes coming from different individuals are paired up. Each gamete carries its own combination of genetic material. This further increases the genetic diversity of the offspring. We previously saw how clonal reproduction transmits the entire genome as it is from parent to offspring. An exception to this are mutations that can occur in an individual and thus modify its genes. If a deleterious mutation occurs in a clonally reproducing individual, it will also be transmitted from parent to offspring. Because the DNA polymerases of all organisms are prone to errors, mutations regularly occur in organisms, and more often they have deleterious than positive effects on fitness. If an organism reproduces exclusively clonally, these mutations will continue to accumulate in subsequent generations. Otherwise put, the mutational load of the clonal organism will increase over time. This process is called Miller's ratchet. A clonally reproducing population can only increase its mutational load, but not decrease it, the same way a ratchet can only move in one direction, but not the other. Therefore, the genetic load of the population will increase under the effect of recurrent DNA mutations, but only to a certain point. If the population accumulates too much genetic load, it will no longer be viable and will start to decline because the individuals will not be fit to survive the selecting pressure acting upon them. Therefore, in a viable clonal population, we expect to have a lot of individuals with intermediate genetic load because of the accumulation of mutations, and only a few individuals with extreme genetic load because if too many deleterious mutations accumulate, the individuals are eliminated by selection. Over the course of the time, if the mutation load is not alleviated, the offspring will accumulate more and more mutations, eventually leading the population to a mutational meltdown. All individuals would have accumulated too many deleterious mutations for the population to be viable. In sexually reproducing organisms, however, thanks to DNA recombination, the ratchet is transformed into a gear. Mutations can be gained, but also lost. Thus, an individual carrying different deleterious mutation on one chromosome can produce a chromosome without mutations thanks to the genetic recombination. This chromosome can be further transmitted to its offspring via sexual reproduction. Most sexually reproducing plants do so via the generation of propagules, such as spores or seeds. The seeds obviously protect the fragile plant embryo from the external environment, but they also have some other advantages. One of them is the prevention of pathogen transmission to the offspring. In clonal species that reproduce vegetatively, pathogens can be transmitted from the parents to the offspring through their conductive tissues, or simply because the parents and the offspring are physically connected to one another, so the pathogens can crawl along them. Seeds, however, rarely have any pathogens inside, with an exception of few very specialized taxa. Therefore, reproducing via seeds allows the population to have offspring with a clean health bill at every generation. Of course, reproducing via seeds enables the plants to disperse long distance within one generation, something that vegetative reproduction cannot achieve, or only after patiently spreading its clones further for many generations. A lot of seeds and fruits have evolved special dispersal structures, such as the eliosomes that favor ant dispersion, explosive fruit capsules for mechanistic dispersion, or pappus extension of the seeds which allow dispersion via wind.